Hang on, let me mute it. Okay, I'll start again. Thank you. Okay, so my name's Louise and I'm an ecological modeller at the UK Centre for Ecology and Hydrology. So I'm relatively new to Phytophthora. I started a postdoc in 2016 working on a UK project called Phytothreats, which was led by Forest Research. And we were trying to understand future global threats to the UK from Phytophthora. So thank you for the opportunity to be able to share with you the work we did in the Phytothreats project and how we're following up on that work. So our brief in the Phytothreats project was to develop some models to help us predict future invasion threats from Phytophthora. And these models were designed to support horizon scanning and risk assessment. And our approach was to use a kind of global cross-species methods to compare between pathogens that are more or less invasive within the Phytophthora genus, and to see whether biological traits can improve our understanding of these threats. So we started asking whether individual traits were linked to a broader range of regions or hosts being impacted globally. But then we tried to explore different invasion stages in more detail by looking at bilateral trade flows among countries and whether there might be interactions between individual traits and import volumes, which could maybe help us to predict which pathogens were more likely to arrive along these pathways. And we also use global niche models to try and identify the important environmental risk factors for Phytophthora species. So what would favour their establishment in new regions, and to see if this could help us to predict how much of a novel region is at risk. And another thing we hope to gain from this project was to develop some links with stakeholders in policy and practitioners in the ornamental landscaping and other sectors, so that we could go on to develop these models with their kind of input and feedback as potential end users. So that hopefully we'll end up with some useful tools based on these models that can help support plant health decisions. So I'll also tell you a bit about how that's going towards the end of the talk. So horizon scanning and risk assessment are kind of key parts of invasive species management. And this is a kind of overview of how those two approaches link together. So the EU has developed a horizon scanning framework as part of its strategy on invasive species. So horizon scanning focuses on the species that haven't yet established, so it's linked to prevention. And it's a way of kind of rapidly identifying and prioritizing future threats using a systematic examination of the information available. But often there's very little empirical data that we have sort of at our fingertips to assess future threats. So horizon scanning is a way of bringing together the kind of limited information and using expert knowledge to try and interpret it. And the aim is to arrive at a kind of ranked list of species, highlighting those with the greatest potential for future impacts. And this can then be used to prioritise which species to take forward for full pest risk assessment. And then at the risk assessment stage, we want things to become a bit more quantitative, and we want to kind of know the probability that each of these threats will be realised. So that might include things like spatially explicit risk maps or more mechanistic spread models that often form part of detailed pest risk assessments. So Phytophthora are a really good system to explore these global horizon scanning approaches. This is a figure from Peter Scott's recent paper, which shows how Phytophthora species diversity has accumulated over time and how this has accelerated in recent years. So now there's much better molecular tools for detection and increasing numbers of surveys in natural ecosystems. So as of this year, I think there's around 180 or 190 Phytophthora species, either formally or provisionally described, but the true diversity is estimated to be 500 or 600. So the greatest future threat might come from the introduction of a novel species about which we know very little. In the UK, we have something called the Plant Health Risk Register, which has 19 Phytophthora species on it, but knowing how to prioritise these species is quite difficult. And what would be quite useful will be some kind of early warning system, so that as these new species emerge, we can try to predict which ones pose a real threat and which will remain fairly innocuous. Because within the Phytophthora genus, there's examples of both very low impact and very high impact species. So these are a few of the plant destroyers from around the globe. So Phytophthora have hosts in multiple sectors. Phytophthora infestans is responsible for potato late blight and the Irish potato famine, and it's a disease that keeps re-emerging around the world. And the picture in the centre is Phytophthora ramorum on Japanese larch in the UK. And that was a very big deal for UK forestry because losing Japanese larch means that Sitka spruce is now the only commercially viable timber species that will grow in a lot of our kind of acid upland forestry sites in the western UK. And another example from the UK is the loss and decline of juniper due to Phytophthora ostracidri. And juniper is ecologically important in the UK. We only have 3 native conifer species. It supports a lot of wildlife and is a woodland pioneer. And we also know that Phytophthora are widespread in the nursery network. So Thomas Young's survey of around 700 European nurseries, and in that they've reported 49 different Phytophthora species, and 91% of the nurseries they surveyed had Phytophthora present. And then the bottom right image is Phytophthora cinnamomi killing lone manzanita scrubs. And that's in a national park in California where those shrubs are endemic. And it looks as though native plant nurseries producing plants kind of used in these restoration projects is one pathway for introducing new Phytophthora species into natural ecosystems. So we've been trying to kind of develop a trait-based invasion framework for Phytophthora to help us to understand how we might begin to predict these future threats, like the ones we saw on the last slide. So this is adapted from a figure in this paper by Miguel Redondo, and it shows some potential invasion pathways for Phytophthora. And we wanted to try and consider the entire invasion process from a global perspective, starting with how those species might initially become entrained on the pathways within their native range and how they might be transported and introduced and then go on to establish and spread. There's quite a lot of human activity that can potentially move Phytophthora propagules around and initiate the invasion process within the native range. So agriculture and forestry and tourism and construction. And these are the things we were thinking about that might have the potential to transport propagules on vehicles and animals and plants and footwear. Native Phytophthora are also quite widespread in waterways, and this water could be used for irrigation in nurseries and horticulture. So all these activities can potentially move pathogens from relatively undisturbed habitats into more human-dominated landscapes. And this means that pathogens are being brought into regular contact with a whole new set of plants, hosts and habitats, especially plants that are bought and sold in our gardens and public parks. So spillover into the ornamental. I'm moving on too fast on. So spillover into the ornamental and forestry sectors is very likely. And then it's quite easy for them to get entrained in international trade pathways, and this is thought to be the major route for transporting Phytophthora to new biogeographic regions. We were interested in whether Phytophthora traits might mediate their abilities to exploit these different transport pathways and move from one invasion stage to the next. So this paper is a really great example of trait-mediated filtering of Phytophthora species, showing how Phytophthora communities change along a gradient of human management in Sweden, starting from nurseries at the highly managed end through urban and agricultural locations, and ending with relatively undisturbed forests. And only a subset of Phytophthora species in Swedish nurseries were also found in the wider environment, and an even smaller subset of those were found in less disturbed forests. And the species establishing in the wider environment tended to have lower minimum temperatures for growth and the ability to form asexual survival structures. And the same authors also showed that trait differences between terrestrial and aquatic species, Phytophthora, meant that they responded differently to temperature and precipitation gradients in Sweden. So we wanted to test whether these trait effects also hold at the global scale, and whether trait filtering plays a role at other stages in the invasion process. And so the team who compiled a species level trait database for 179 Phytophthora species are shown in these photos, and also a complete ITS phylogeny. There's a lot of morphological and physiological data available for Phytophthora, and these data become available fairly quickly once a new pathogen has been isolated, which is good, but historically those traits have been measured for morphological identification. So we know very little about their function and adaptive value. So we made some hypotheses about how these traits might map onto the different pathways and invasion stages. And to try and capture patterns of invasion and impacts, we also collated as much data as we could find on the distribution and host range of different Phytophthora species across these different habitats and sectors and climatic regions. And there's more than 100 pathologists on multiple continents who contributed to bringing together distribution and host data for Phytophthora from nurseries and gardens and agriculture, horticulture, forestry and natural environment. And this means we can examine the global distributions of 173 Phytophthora species at country level, looking at where and when species have been detected for the first time. And we also have some finer grain data. So this is around 27,000 georeferenced occurrences to help explore the environmental risk factors for establishment. We initially used the distribution and host database to find some coarse metrics of Phytophthora global impacts. And these metrics were designed to roughly capture some different aspects of kind of invasion. So the number of countries reached was chosen because most pest and pathogen reporting is done at the national level. And these might come from plant health surveillance in nurseries and the wider environment. So this seemed like the most comprehensive picture of the extent to which Phytophthora species have been transported globally. But the number of countries reached doesn't really get at the distribution of those countries and whether they're similar or very different climatically. So we also extracted the known latitudinal limits of Phytophthora species from the global data that we collated. And then neither the number of countries reached or the latitudinal limits can tell us much about the damage caused by those species. So we also counted the number of host plant families known for each Phytophthora species. And we wanted to identify whether traits could be useful predictors of any of these impact metrics. And so these are some results for models linking traits to the number of countries reached. And Phytophthora species traits and phylogenetic position explain between 21 and 25% of variance in the number of countries reached by those species. We used a model comparison approach here where we fitted a model for every possible combination and subset of the trait predictors. And the reason for doing that is it's a good way of dealing with any collinearity between traits because it allows you to see whether including or excluding other traits from the models kind of changes the effect of sort of the focal trait that you're interested in. And we ranked these models from best to worst based on their predictive power using cross-validation. And this figure shows the subset of the best performing models. So each row is a trait effect and each column represents a different model. So the redder colours are positive effects of traits and the bluer colours are negative effects. And the depth of colours shows the effect sizes of those traits. So where you see bands of stronger colours, that suggests there's a consistently strong effect of that trait across all the best models. And the first thing to note is that the number of countries reached most strongly depends on how long it's been since that species was described. This is a covariate that tries to adjust for how much knowledge we've accumulated about that species, but really we're interested in what the biological traits can explain, how well the biological traits can explain the remaining variability in species impacts. So we found species with lower minimum temperatures for growth were those that have been reported in more countries. So this is consistent with cold tolerance being linked to Phytophthora species establishment outside of nurseries in Sweden in those studies that I mentioned by Miguel Redondo and colleagues. There's a caveat there, which is that it's possible that our impact metric is biased by more intensive sampling in northern temperate regions. And perhaps if tropical regions were equally well sampled, we wouldn't find the same result. And then other traits that were important were the ability to infect roots, which might be an indicator of kind of more soil-borne life history and maybe a strategy that makes transport with ornamental plants more likely, and maybe even evading detection in transport more likely. And then there's two traits that seem to show some collinearity. So when Caducus sporangia are in the model, foliar disease tends to be absent and vice versa, suggesting there's some redundancy there. And those two traits are probably associated with an airborne dispersal strategy that might facilitate more rapid aerial spread among susceptible hosts. And traits were able to explain slightly more variance in latitudinal limits, so around 40% in the best performing models. And cold tolerance was again a good predictor of which species were being recorded at higher latitudes. And latitude was intended as a proxy for these kind of larger scale climatic gradients in temperature. And it does seem plausible that cold tolerance is adaptive for establishing in a broader range of climates. It's also kind of interesting given the surveys being carried out to look for the geographic origin of some of these invasive Phytophthora. And those surveys have identified high altitude regions, including the Himalayas and Taiwan, as kind of possible centres of native Phytophthora diversity. And I think Phytophthora remorum has now been found in natural ecosystems in Japan. So it's perhaps those species that are successfully transported and then established at higher latitudes are those that are kind of pre-adapted to cooler regions in their native range. And then the absence of oospores was also a good predictor of which species were reported at higher latitudes. And this is a characteristic of many clade 6 species found in streams and waterways. And in the studies in Sweden, waterborne species were less sensitive to the temperature gradients than soil-borne species. So it seems that cold tolerance and the absence of oospores might represent two different strategies for being successful at higher latitudes. And they certainly don't seem to be correlated effects. But the value of oospore absence in these kinds of trait-based invasion models might be quite limited if it captures the potential distribution of native or naturalized pathogens that are generally a bit more opportunistic and quite low impact. And then species traits and phylogeny explained around 31 to 34% of the variance in host plant families. And root and foliar disease symptoms were both present in the best performing models again. So perhaps this captures something about the detectability of those species on their hosts. But other traits that were important were a bit different for host range. So species with thicker oospore walls were associated with infecting more host plant families. And if thicker walled oospores kind of do remain viable for longer in the soil, then this could increase encounter rates and maybe successful transmission between novel hosts, which might increase the chances of host jumps maybe. And then faster growth rates were also associated with more host plant families. So maybe faster growth rates could be helpful in adapting to new hosts rapidly if it gives species a kind of competitive advantage when colonizing host tissue. And maybe if it translates to faster generation times and transmission between new hosts. And then we can also compare the observed and predicted impacts from these trait-based models to try and identify those species who have reached a lot fewer countries than the models would have predicted based on their trait values and phylogenetic position. This is a list of Phytophthora species first described in the last 10 years. And here we kind of use the trait-based models to rank them in order of their potential threat based on which have been reported in a lot fewer countries than our models would predict, given the kind of similarity of their trait values and their close phylogenetic relationships to other high impact species. So these ranked lists could be a good starting point for the horizon scanning type exercises that I sort of described earlier. But they really need some scrutiny by expert pathologists. So incorporating them into this kind of expert elicitation and consensus building process that's used in horizon scanning could be quite productive. The list would then get moderated and revised based on their expertise. And then maybe the model could be refined based on some of this feedback and then go back into the process. So through a few iterations, we can arrive at some kind of semi-quantitative ranked lists of pathogen traits by integrating these cross-species models with expert knowledge. And next, we wanted to build on the trait-based approach by developing some models that could tell us more about where these species might be more likely to reach and whether traits play a role. So the objective here is moving a bit closer to risk assessment. And to do this, we wanted to look at patterns of new Phytophthora arrivals globally. So we just defined a source range for each species as countries where the species was already present prior to 2000. And this is what that would look like for Phytophthora remorum. And then after 2000, any new detections outside of the source distribution were classed as arrivals, which are green, and then non-arrivals, which are shown in blue. And this time we wanted to build in the kind of major pathways for transporting these species globally. So we used live plant imports from potential source countries as a measure of trade connectivity. To estimate probability of arrival, we used a model which allows Phytophthora species to have different responses to trade connectivity and to climate matching. And the interaction terms between traits and connectivity to source regions would then capture how much of the species level variability is due to trait differences. So we're trying to understand whether particular traits can mediate how well Phytophthora species can exploit those global trade networks. We also tried to do a better job of accounting for the spatial patterns of Phytophthora reporting this time, because new Phytophthora arrivals will probably be very biased towards the wealthier countries with more resources for plant health surveillance and research. So the probability of arrival will vary between countries depending on kind of general biosecurity capacity and the level of reporting carried out by national plant protection organisations. And we use data from the EPO reporting service archive as a measure of plant health surveillance for each country. And this measures the number of new reports of any plant pest and pathogen. But we also wanted to try to account for more general biosecurity effort at the country level. So we included these two metrics that were presented in this Nature Communications paper. And Reagan Early supplied this data. And these metrics come from scoring how well countries have met targets for enacting legislation and other preventative measures for invasive species spread. And these targets are set out by the Convention on Biological Diversity. And these are some early results. So we're finding that more imports from the source distributions increases the probability of a new Phytophthora detection. But the strength of that effect seems to vary among species with different trait values. So the different coloured lines in this figure show the responses to trade among species with broader, median and narrow thermal tolerance ranges based on their growth curves in the trait database. The species with broader thermal tolerance ranges are more likely to arrive as input volumes increase. So it's possible that broader thermal tolerance ranges allow species to tolerate the kind of more extreme and variable conditions during transport, but it's not clear from our results on the analysis whether this trait's more important at the transport stage or the establishment stage, or for both. So climate matching within the source region significantly increases the probability of a new Phytophthora arrival, but we haven't yet looked at whether thermal tolerance traits interact with this effect, and if so, that might imply that this is a trait that favours establishment more than transport. And the next analysis we did was designed to produce suitability maps based on environmental risk factors that could indicate how much of the UK might be at risk from particular Phytophthora species. So to validate this approach, we tested how well these global niche models could predict the suitability in the UK using Phytophthora species that are already present here. So we left out the UK records when fitting the model so that we could use them to test how well the observed distributions match with the predicted suitability. And if this approach works, the idea is then that we could create risk maps for species that haven't yet arrived in a particular region. However, we found there were only 9 species in our distribution database with enough occurrence records to make kind of more robust niche models. And all of them were already present in the UK. So we can kind of test these approaches, but it's not really possible with our current data to produce risk maps for Phytophthora species that haven't arrived yet. And to help us with these global niche models, we kind of cornered around 40 pathologists at one of the Phytothreats project meetings, and gave them kind of post-it notes and breakout groups and picked their brains about potential risk factors for Phytophthora establishment. And we ranked the risk factors based on how many times they were identified by the participants. And then we had to select some suitable global environmental layers to use as proxies for these risk factors. And the risk factor mentioned most often was the distribution of susceptible hosts. So we used global data on land use to try and derive kind of layers of forest cover, agricultural cover, and urban cover. And urban cover was meant to capture kind of epicenters of outplanting of ornamental hosts. And agricultural cover was meant to represent the distributions of horticultural and agricultural hosts. Forest cover was chosen to capture the availability of woody hosts, so more semi-natural and managed forest-associated species. And then we selected 4 climatic variables as proxies for the climate risk factors that were identified. So we extracted a moisture index based on evapotranspiration and rainfall deficit as a proxy for wetness and standing water. And then mean summer temperature and the minimum winter temperature we chose as variables related to Phytophthora species thermal niches. Then the seasonality of the models was also mentioned by a few participants. So we included precipitation seasonality to see if drier periods could restrict the distributions of some Phytophthora species. And it's quite reassuring that the climatic limits we inferred from global occurrence data were quite well correlated with species temperature growth curves documented in the trait database. So the correlation between highest summer temperature where a species occurred and that species maximum temperature for growth in the trait database was about 0.6 across the 9 species. And then the lowest winter temperature where a species occurred in our global database correlated quite well with that species minimum temperature for growth. So the models are making hopefully fairly accurate assumptions about the thermal niches of these species. And in those niche models, climatic variables had a greater influence than land cover on global distributions, with the strongest effect being from winter temperature and the seasonality of precipitation. There were slightly weaker effects of summer temperature and the moisture index. So the fact that land use variables are coming out as less important than climate suggests that using these kind of land use types as proxies for host distributions is probably not doing a very good job of capturing the co-occurrence of Phytophthora species with their hosts. And these maps show how we evaluated these global niche model approaches. So the global map shows the occurrences from outside the UK that we used to train the model and the predicted global suitability for Phytophthora ramorum. We used a metric called AUC to measure the predictive success of the models. So for this, a value of 1 means perfect discrimination of presences and absences, and a value of 0.5 means the model's kind of no better than random. So global predicted suitability matched very well with the known global distributions of species. And using cross-validation with subsets of the global data, the AUC was greater than 0.97 for all 9 species. But what we're really interested in is how well these models can predict and extrapolate beyond the known ranges of Phytophthora species, so kind of into novel regions. And the UK maps show that the UK suitability predictions from those models, and the black points are the validation records. So these are just limited to natural and outdoor managed environment, and the regions with higher predicted suitability do seem to broadly match with the known distributions of Phytophthora ramorum in the UK. And the AUC is 0.74, and across the other species, the other 8 Phytophthora species, the AUC exceeded 0.65 for all species except for Phytophthora gonopodiades. So our overall conclusion is that global niche models could provide useful risk maps for Phytophthora species establishment in regions beyond their known range, but that the kind of incomplete and extremely biased recording that we have, that we've kind of collated globally, is a major challenge for this type of modelling. So investing more in these kinds of centralised databases for pathogen distributions could be quite important for developing these approaches a bit more. And then for the final part of this talk, I'm just going to describe how we've been engaging with some potential end users for these models. And so at one of the Phytothreats stakeholder workshops, we got a chance to demonstrate some prototype tools based on the models and data. And there were some really useful discussions, which made us think we should try to capture some of this information in a kind of more formal and structured way. So we now have a kind of small role in a project funded by the Scottish Plant Health Centre, and it's led by Mariella Mazzano at Forest Research UK, and it's predominantly social science in focus, And it involves mapping the stakeholders that play a role in large-scale planting projects, so specifically infrastructure projects like road and railway construction, which involve quite a large landscaping and planting component. And also planting for environmental benefits, including biodiversity and carbon sequestration. And the kind of overarching aim is to understand the opportunities and barriers to minimising plant health risks to Scotland within these different sectors. And our role within the project is to kind of develop and present some interactive tools based on our models to these key players in landscaping and environmental planting. So we have 2 online workshops organised for later this month with representatives from these larger organisations who have a role in kind of choosing the sites and procuring the plants for these projects, or maybe to provide plant health advice to them. And we wanted to try and understand what sources of plant health information are already used, and do the needs for plant health information vary between sectors, and what information about plant health is lacking that could help with their planting decisions or advisory roles. And then how could we improve the tools we've developed? Do we need to tailor them to particular pathogens or environmental context? And then during the online session, they'll be able to kind of access a set of tools that we've sort of that we've based on the outputs of our models, and interact with these data and maps that we've created. And they'll also complete some short questionnaires about different parts of the tools so that we can try and identify any common themes in how these models could be of use and where they need to be developed further to better support plant health decisions. So it's a bit beyond me to figure out how to embed a video where I could kind of try and demonstrate some examples of the tools we've developed. So I'm afraid these are just a series of screenshots. But if you want to have a play yourself, the link is there at the bottom. But you have to bear with me because they're not finished yet. But hopefully they will be in the next couple of weeks in time for the workshops. And this is what the set of tools looks like so far. So it's a kind of collection of web pages where you can visit different help from the Phytothreat project. And there are, I think, 4 sections in various stages of completion. And the first is where a Phytophthora are coming from, which takes us to a kind of interactive global map, and it's designed to help users explore the risks associated with importing plants from different regions globally. So in this box in the top right, you can toggle between risk factors associated with new arrivals of Phytophthora pathogens. And you can choose to look at how much Phytophthora diversity there is in different regions and what the annual trade flows are alike from those countries. And because this is for the Scottish government, we're assuming the UK is the importing country. But the tool can be set up to do this for any other importing country. And you can also map the climate similarity between where you are and the rest of the world. And you can also map exporter biosecurity capacity based on compliance with invasive species targets. So that was the data from Reagan Early's paper that I mentioned. And then you can combine all of these risk factors into a kind of composite export risk for each region based on trade model predictions. And then if you click on a focal country, you can view a kind of pop-up table listing the Phytophthora species that have been reported there. So if we were considering importing some trees from Australia, I would get this table. For the species that aren't already present in the UK, it will give you an estimate of arrival risk for each species. And that's based on the thermal tolerance traits we included in our trade models. And then we can also explore the outputs of the niche models and view the climate similarity for different Phytophthora species across the UK. And you can toggle between suitability for different species using the button on the right, and then zoom in to see local site level suitability. So maybe this could help practitioners to think about whether their planting sites might be vulnerable to particular Phytophthora disease outbreaks. And the black squares are the known occurrences of Phytophthora outbreaks in the wider environment. So you could also kind of evaluate how close your site is to other sites of known infection. And if you click on those Phytophthora records, you can see the host or the hosts that were affected in those outbreaks. So you could maybe consider also whether your proposed planting scheme includes susceptible hosts. And then to summarize, these are some of the insights we gained across these different trait-based analyses of Phytophthora invasion threats. We found that risk of arrival is a function of live plant imports from the source ranges of Phytophthora species. And that climate matching was also important in explaining new Phytophthora species or new Phytophthora detections. But the importance of trade connectivity and arrival varied across Phytophthora species, and those with broader thermal tolerance ranges were more likely to arrive with increased trade. And we found that the suitable area for Phytophthora species within the UK could be reasonably well predicted by matching their global distributions to climate and land use predictors, particularly winter temperature and rainfall seasonality. And that species thermal niches were quite well captured by their temperature growth curves obtained in the lab. So it seems like this could be a potential way to assess how much of a novel region might be at risk from Phytophthora species that haven't arrived yet. And we found that species traits and phylogenetic position did have some power in predicting future global impacts. So cold tolerant species reached a greater number of countries and higher latitudes. And species with faster growth rates and thicker oospore walls tended to infect more plant host families. But we do need to find ways to better capture these global pathogen data across borders and sectors and disciplines. So our data are quite biased towards northern temperate areas, and we do need more distribution and trait data from other climatic regions to kind of really understand the sort of nature of trait filtering in different regions and how this influences future threats from Phytophthora. And then to finish up, these are kind of some of our proposed next steps. And one area that could be really fruitful for understanding Phytophthora invasions is how rapidly these pathogens could evolve. So trait-based approaches I've described sort of here assume that these traits are quite static and there's very little variability within species, but we've done some phylogenetic analysis as well within the project that shows that some of these traits have been quite labile through evolutionary time. And the human-mediated pathways that enable Phytophthora invasions also potentially impose some quite strong selection pressures on the pathogens. So it would be quite exciting to explore whether human influences during invasions might be kind of playing a role in these trait shifts that might then exacerbate the spread and impact of Phytophthora. And there might also be different strategies for being a successful invader. So it would be good to test whether there are combinations of traits or kind of trait syndromes arising from those phylogenetic relationships. And perhaps they could be more useful than the individual traits in capturing successful invasion strategies. And so I think that's all. Thank you to all my co-authors and the funders of the Phytothreats project. Collating the Phytophthora trait databases was a huge collaborative effort, and there's over 100 people who I'm very grateful to for sharing their amazing data sets. And we're also very appreciative of the stakeholders who've provided feedback on the modelling tools already, or who will do later this month. And so thank you for having me, and sorry about the delay. I've put you all off schedule, I think.